Hi, everyone. Thanks so much for this opportunity to give this talk today. I'm José Rafael. I'm a postdoc researcher at the University of Reading. But before to came to the UK, I was work in Universidade Regional Amazônica ICANN, which is a university based on the Amazon jungle. Basically, ICANN means jungle in the Shuar language, the local language. So that's why today I'm going to talk about my favorite snake, which are the Ecuadorian coral snake venoms. But first of all, allow me to introduce Ecuador, because since I came to the UK, when I'm telling people I'm Brazilian, but I was living in Ecuador, everyone thinks that Ecuador and Brazil are the same place. They don't know Ecuador. So Ecuador is a South American nation, which is well known for having the highest number of reptiles per unit area. Um, it has diverse ecosystems, which are home to 240 snake species, and including 37 venomous snakes. And Ecuadorian snake venoms have been neglected for such a long time. When I just went to Ecuador 6 years ago, only few viper snake venoms have been characterized. So in terms of snake bites is still a complex public health problem in the country. Ecuador experienced an incidence of 1,400 to 1,800 snake bite cases annually. The most affected areas are in the Amazon regions, where we don't have really actually good infrastructure, healthcare systems. And who dies from snake bites in Ecuador? Basically, as similar to other tropical countries, people, indigenous peoples, and low-income agriculture workers. And uh, it, when it comes to the treatment, we are facing many challenges. We don't have local production of antivenoms. I mean, Ecuador relies on imported antivenoms, and there is a significant gap because the country, uh, the government only import antivenoms to treat or manage bottropic envenomations. So, I mean, we don't have coral snake antivenom available to treat those envenomations. So when you go to the hospital, if you get bitten by coral snake venoms, there's a lack of training as well. All doctors and physicians, they are treating coral snake envenomation use bottropic antivenoms. And of course, the mortality that you got in the country is basically caused by coral snake venoms. That's why it's really important to study coral snake venoms there. But when I went there, one interesting as well, we didn't know the composition of coral snake venoms in Ecuador. Although you have 19 snake species, there is no proteomic data available for coral snake venoms. If you look at this map, many of the countries producing local antivenoms, they have already characterized their venoms. And which is interesting is, as you know, viper venoms and elapidie venoms, they are quite different. Elapidie venoms, they are usually composed by low molecular weight proteins, and it's basically a combination of three-finger toxins and phospholipase A2. And interestingly, there are two compositional partners. We got some microurus venoms that contains a lot of phospholipase A2, while another coral venoms, they are dominated by three-finger toxins. And what is even more interesting here, if you look at this map again, there is a trend. You can see that venoms from North America, they tend to express more phospholipase A2, while venoms from South America, they are basically dominated by three-finger toxins. But if you see here, Ecuador, is almost in the middle. You don't know until recently nothing about the coral snake venoms here, and is I would say, a paradise for toxinologists. So it's not only a beautiful place to live, but also is an ideal place to study this snake, because the country is divided by the Andes Mountains. So you have completely different ecosystems and regions. So of course, when you look the proteomes profiles available for coral snakes, uh, uh, basically the studies comes from Calvet and Bruno Lamont group. Around of 22 snake venoms were characterized, but of course we need much more studies to confirm this trend. And in addition, there are some studies demonstrate that this dichotomy or polarization of the venoms have some impact on antivenom efficacy. There is this study from Lomont and collaborators that illustrate this really well. They have evaluated the ability of a coral snake venom produced in Ecuador to neutralize several coral snake venoms. What they found was that the antivenom produced using micronigro, which is a PLA2 dominant venoms, effectively neutralize those venoms that contains high levels of PLA2. But this antivenom struggles to neutralize three-finger toxins predominant venoms. So motivated by these challenges, we decide to start study the Ecuadorian snake venoms. So initially, we are start to study two medically important coral snakes, Micrurus mipartitus and Micrurus hilari. As you can see here, they have different geographic distribution. Micrurus mipartitus is found in the coast area of Ecuador, while Micrurus hilari is found in the Amazon region, really close to where we are based there. So to understand the venom composition, we applied the venomics approach developed by Professor Calvet. And what we found, as you can see here, these two venoms, they have different biochemical profiles. Micrurus hilari has prominent peaks in the middle of the chromatogram, while Micrurus mipartitus, the most intense or prominent peaks are in the first third of the chromatogram. And if you look at the SDS page profiles, it has basically low molecular weight proteins, and overall, our proteomics analysis have confirmed that these venoms have the molecular signature of coral snake venoms. They are basically a composition of phospholipase A2 and three-finger toxins. And interestingly, we found both venom phenotypes in Ecuador. Venoms from the Amazon area tend to express more phospholipase A2, while venoms from the coast, they have more three-finger toxins. And these profiles also aligns with previous studies of other groups using these snake venoms from this snake species from other countries, such as Colombia and Costa Rica. So the first lesson that we learned from this study is in Ecuador, you have the coexistence of both phenotypes, PLA2 rich venoms and three-finger toxins dominant venoms. And this highlights the possible need of antivenoms able to recognize both venom profiles. But we still need more data to confirm this trend in the country. So that's why we decide also do the proteomic and the functional study of Micrurus ornatissimus. Micrurus ornatissimus is a snake found in the Amazon area. So as expect, when you did the proteomic analysis, we found, which was expecting that these venoms is dominated by phospholipase A2. And due to the absence of lack of antivenoms in the country, we also decide to screen the small molecular inhibitors because it's really nice. Every conference that you come here, people are trying these inhibitors for ALAP venoms in Africa, but these are few studies with microrurals. So when you have done the screen, you have seen that variscladib and marimastad and pranomastad, phospholipase A2 inhibitor and MP inhibitor, they inhibited the enzyme activity of the Venoms enzymes found in this venom. So as you can see, this venom contains a lot of PLA2, and you have clinical report that it can also cause muscle damage. Although the neurotox manifestations are the main clinical relevant, we decide to study the myotoxicity induced by this venom. So we start to see the cell viability using the colorimetric MTS assay, and what we found is this venom is able to kill or reduce the myoblast viability. And when you tried the different inhibitors, as you can see, the inhibitors are alone, phospholipase inhibitor and MP inhibitor. They don't have any bad effects to the cells. However, when we incubate or pre-incubate venoms with with PLA2 inhibitors, you have an increase in the cell viability. However, it's not 100%, although we have increased the concentration of variscladib. And marimastad does not affect. This data, what's telling for us is basically the myotoxicity caused by macros or natissimus seems to be due to PLA2s and maybe a synergy of some L-amino oxidase that we also found in that venom. That's why we don't have 100%, but the MPs, they don't seem to play a role here in the myotoxicity. So we also studied the impact on fully differentiation myotube. As you can see, in the muscle cells, you have myoblasts, and you can have also the differentiate myotubes. And basically, we stain for myosin heavy chain. Without venoms, you can see long and beautiful health myotubes, but when you treat the venoms with different concentrations, when you treat the myotubes with different concentrations of venoms, basically the venom is destroying the myotubes. What is interesting here as well is the venoms is killing myotubes in lower concentrations than myoblasts. If you look at the myoblasts, you have here, after 50, you have a more significant reduction in myoblast viability, but in the case of myotubes, we need lower doses, which means that the myotubes, they seem to be much more susceptible to venomous enzymes than the myoblasts. And since you have already seen that variscladib can neutralize PLA2 enzymes, you decide to repeat these experiments pre-incubate with variscladib. And as you can see here, when you pre-incubate venom with variscladib, you can see here again the myotubes, which means that targeting PLA2 can be a good alternative to manage the myotoxicity caused by these venoms. We have also studied the effect of these venoms in the myoblast fusion, in the differentiation process. As you can see here, when you incubate myoblasts with the venoms, this impairs the multi-step process of differentiation. You cannot see the formation of myotubes. However, again, when we pre-incubate with variscladib, we have seen the formation of these beautiful myotubes. So we also have explored new avenues to see if these venoms also can cause some blood disturbances. So you have analyzed the impact in the extrinsic and extrinsic pathways using coagulation assays and route analysis that people have previously explained for us in this conference. And as you can see, microdose venoms seems to extend the clotting formation times in all of our assays. Then, as these venoms also contain MPs, you decide to evaluate the fibrinolytic activity. As you can see here, microdose ornathissimus are able to digest the alpha chain of fibrinogen, similar to another microdose venomous previously report. But when you pre-incubate this venom with MP inhibitors, you have seen intact alpha chain fibrinogen. So we also have studied the effects on human red blood cells. As you can see here, this venom seems not to affect the human red blood cells, something that also have been explored for Gutierrez grupos, showing that dogs and mice, they seem to be more susceptible. And this is maybe due to the different proportion of phospholipids in the red blood cells. So to sum up, here you have some take-home message. We have provided 3 new proteome profiles for Ecuadorian pharaoh snakes. So we are adding proteome pieces and also some functional data to the knowledge about the divergent profiles of corals. I think maybe this is an important step for Ecuadorian contest. And we are working closely to the government now to establish policies to have antivenoms to neutralize microdose envenomations. Our results also showed that small molecular enzyme inhibitors, they may be useful in mitigating the toxic effect caused by microdose, but of course, we still need to move to in vivo studies. All our assays have been pre-incubation, so there's some challenges to translate this data. But our pending task here, and really important, really needs immediately to try to see the neutralization of these venoms using available antivenoms from Costa Rica, Brazil, and Colombia to help the country to take decisions and which antivenom can be more useful in the case of these 2 profiles. Finally, I would like to acknowledge Professor Varapuri. I have been working here in the UK for 2 years, and also my students from Icahn University, and also my collaborators, especially the Professor Bruno Lemont, who has helped us a lot with the proteome analysis. So yes, thank you for your time and attention. I'm happy to take questions of the audience.